Hello, now we're on the introduction to chapter 16. This is all going to be about the sense organs, but first we're just going to do a little bit of introduction to pain. How do we feel pain? What are some of the other receptors that can feel chemical, that know when the chemicals in our bodies change, like pH and stuff like that, and also when pressure, the baroreceptors that are going to be important for blood pressure and other types of things with the blood and circulatory system. So it's important for us to go over all these different types of sense organs before we even get to taste and then smell. We're going to do taste and smell together with pain because hearing in equilibrium is pretty long and vision is extremely difficult just because there's a lot of steps on getting photons of light into pictures that we can kind of interpret. So let's get started with the introduction. So the sensory input is vital to the integrity of personality and intellectual function. Information communicated by the sense organs never comes to our conscious attention because we don't really even think about it. We don't really have to think about, hey, I think I'm going to be cold. I need to warm myself up. Hey, I think the vessels in my body are going to are higher. So I think they're having a lot of pressure. So I should probably go do something about that. Most of these things are just done by our body. We don't have any control over them. And that's good because if we're sleeping and our muscles are being are under high tension, it would wake us up. If we're cold or if we're warm, our body can just react to it immediately once it feels that type of input. So our sense organs initiate somatic and visceral reflexes. We kind of went over a few of those in the reflex lecture, so we're not going to really go over them again. And these are going to be indispensable for homeostasis and our survival. So first, what are the different types of receptors we have? Phasic and tonic. Phasic is pretty interesting because it generates a burst of action potentials when first stimulated. And then it quickly adapts, and then it sharply reduces or stops even signaling, even though the stimulation continues. Think about smell. Smell's the big one. It's underlined right here because we've all done this. We've all taken a shower. We all had to go outside. We were going to go on a date. We're going to go to the movies. We're going to go hang out with friends. We're going to go, meet people over at a house. And what we do is we take a shower. We might use scented soap. We might use cologne or perfume. We might use special lotion. You put it on at first. It smells very intense. And then pretty much by the time you get ready and you go in your car and you're leaving, you don't smell it anymore. That's because the sense of smell is phasic. So what happens is right when a new scent or smell hits the olfactory nerve, it's very, very high. Sends a signal to the brain that says, hey, something new is happening. Then after a while, it doesn't become as important, so the brain just kind of starts to turn it off. And it does this because your sense of smell is there for danger or new or novel sense. So unless a new thing kind of comes into your, passes by your olfactory nerves, it's not going to signal again. That's why some people, when they put on perfume or cologne, they might say, oh, I don't smell it anymore. It must have wore off. They put some more on, and then that'll wear off. They put some more on. But every time you're putting more and more perfume or cologne on, you're just becoming stronger and stronger scented to the people around you. Also, hair movement. You want to see, if the wind's coming a certain direction or if your hair is moving in one direction, you want to feel it, but you don't want to keep feeling it because nothing new is changing. And also skin pressure. So if you feel pressure right away, it's like if you put on a shirt that's too tight. You put it on, you're like, woof, that's too tight. But if you leave it on, eventually you won't feel that shirt's tight anymore. The other one is tonic. And tonic receptors adapt slowly. They generate nerve signals more steadily and eventually will reach a point where you have to make a change. And these are going to be proprioreceptors, body position, muscle tension, and joint motion. And we've all been either running, exercising, doing different physical activities where, let's just say you are holding on to a bar. you're in elementary school or middle school, you're doing that presidential fitness test, and you're hanging onto the bar, and you have to make it to a minute to get a certificate that may or may not be signed by the president. And you're hanging onto the bar, and after a while, your muscle fibers are stretching so much, there's so much tension in those muscles, your fingers are going to let go. You don't consciously say you're going to let go, but your body just says, that's enough, I'm done. Also, body position. We talked about these different types of receptors when we talked about different types of reflexes in previous chapters, but what if you're falling asleep in class? Your body position is going to change. You're going to wake up and you're going to readjust your body position. And also joint motion. You do not want your joints to go too one way or to the other way, because then stuff can pop out of a joint. So after a while, if too much pressure is put on a joint, those muscles will just stop to work and hopefully save the joint until you really damage it. So that's why tonic builds up slowly until it reaches a point where you have to stop. Phasic starts off very, very strong and then goes down over time. So we can have different types of classifications. By modality, pretty much what they do. Where's the origin of the stimuli? Is it outside or inside? Or by distribution. Really what we're going to see is thermoreceptors pretty much respond to heat or cold. Photoreceptors are going to be the eye. It's going to be the rods and cones, because photons of light are going to activate them. Nociceptors are pain. We're going to talk about them later. So that's not in red now. And chemoreceptors, chemicals. Most likely, the main one that we're going to talk about is going to be the sense of smell, including odors, but also when we talk about the circulatory system, how the blood is so intertwined with the acid and base balance of blood. And also mechanoreceptors. Is there some type of mechanical action? And this is going to respond to physical deformation of cell tissue caused by vibration, touch, pressure, stretch, tension. These are all going to be the ones that are going to be, it's stretching, there's too much pressure, something's moving. These are going to be mechanical because something mechanically is happening to that receptor. And like I said, it can come from outside, it can come from inside, and proprioreceptors is body position and movement. Also, they can be general. They can be widely distributed, or they can be very, very, very special, which vision, hearing, equilibrium, taste, and smell. The special senses is what we're going to talk about in this chapter. So, what does it mean to be unencapsulated nerve ending? It just means that the outside is not wrapped in connective tissue. So, free nerve endings are just that. They just go through an area or tissue and they're just unencapsulated. They're there for pain, temperature. You want pain and temperature to be very, 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 very sensitive. So it's on the skin and mucosal membrane. So if pain or temperature changes, we can sense that really quickly, and we don't have any type of membrane that is wrapped around it to actually dampen or insulate those feelings coming into the nerve endings. They also have tactile discs, where you can see the tactile disc is going to be right on top, and it's for light touch and texture, often associated with the Merkel cells at the base of the epididymis. We talked about the Merkel cells way back in the integumentary system in the skin. You may or may not remember it, but either way we've talked about it, so it's not going to be read now. We also talked about the hair receptors and monitor the movement of hair. We talked about that too when we talked about skin. We also talked about the dendrites wrapped in glial cells or connective tissue. And this is kind of important, but some connective tissues can enhance the sensitivity or selectivity of response. We're not going to go into those types of receptors that use connective tissue, but you will see some in AMP2, so you will see some wrapped in connective tissue that enhances the selectivity of the response and actually changes how that receptor works. So we talked about tactile mesenteric corpuscles, these in the dermal papilla. We talked about those in the integumentary. We didn't talk about these, but these are tactile in the mucous membrane, and you will kind of hear about these when we talk about the respiratory system. The lamellar or the Pacinian corpuscle, These are going to be the pressure receptors in the skin that we talked about. And then the bulbous Rufian corpuscle are going to be tonic. These are going to be heavy touch pressure of the skin and stretching. We didn't really talk about those, but this one does very phasic. If you feel pressure, it all of a sudden reacts. This one builds up for joint movements and other stuff. And all of those are going to be encapsulated by a certain type of connective tissue. But again, we went over most of those. You saw that they were covered with something, so I think you can kind of figure out those were encapsulated in some way or another. Now we're on to pain. Pain is a discomfort caused by tissue injury or noxious stimulation. And it always, if it gets to a certain point, will lead to some type of evasive action. If you have a pain, usually you can ignore it for a certain point, but then it reaches a point where you're just like, I need to get out of that situation. And it's important because it protects us. One of the really bad drawbacks and consequences of diabetes mellitus is you have diabetic neuropathy. And diabetic neuropathy is when you lose the ability to feel pain. So people who have diabetic neuropathy, they should be monitored at all times if they put a cold pack on their foot, if they put warm packs on their legs. If they have muscle injury, you might want to put heat. If you have a swelling or something, you might want to put cold. But if you have diabetic neuropathy, you can't feel the pain if your skin starts to die because of the intense cold. So what happens with some of these people is they're not paying attention. They'll look down. They might get gangrenous limbs. They might have issues because they just can't feel the pain. They might be stepping on something and they don't know that they're stepping on something that's cutting into their skin. So with diabetic neuropathy, it can become very bad. And usually at this point, the limbs are the first ones that get damaged because you can't touch, you can't feel your feet. So usually they'll lose their legs or lose their limbs because of diabetic neuropathy and also because of poor blood circulation. And I mentioned this, nociceptors. These are the type of pain sensations, and they're going to have two types. One's going to be fast and one's going to be slow. Fast, we've already learned this, are always going to be myelinated in the body. Myelinated, and slow is always going to be unmyelinated. So it goes back to what we originally learned with neurons. If it has myelinated and it has a large diameter, fast. If it unmyelinated and smaller diameter, it's going to be slow. So these are going to be those types of receptors. You can also have somatic pain from skin, muscle, and joints where you don't really know where they're coming from. Visceral from the viscera is going to be the one that's poorly localized. So the difference is a bug lands on your skin, you can feel where that bug is. You have something happening in your body coming from the viscera, that membrane that surrounds organs that we talked about. Sometimes it's digestive, sometimes it's poor blood flow, sometimes it's, a bunch of other little things like even a stretch of a muscle. You don't know exactly where it's coming from, but you feel that something's wrong. Some people might say, oh, I've got stomach pains, and actually touching their colon or small intestines. That's why, because they don't really know if it's in their stomach or if it's in their intestines. It's poorly localized. Injured tissue releases chemicals. They stimulate pain. Brandykinins is a peptide that promotes inflammation. It also makes us aware of an injury and activates a cascade of reactions that's going to promote healing. Histamine, prostaglandin, serotonin are also going to stimulate nociceptors. And this is where our body says, hey, we're in pain, we need to change something. And this brings us to referred pain. And referred pain is usually the one where you think, I'm having a heart attack, why is my left shoulder in pain? Because this is pain in the viscera, often mistakenly thought to come from the skin or other superficial sites. So it results from a convergence of neural pathways in the central nervous system. When we did the spinal cord, you learn that it has all of these little roots coming out, and it goes through one pathway between the vertebrae, and it goes out to muscles, skin, It can be sensory, motor, or it can be mixed. The ones that are sensory, motor, and mixed, they keep going up. The brain doesn't know exactly where it's coming from because there's not a lot of connections between the brain and that particular pain. So what happens is you assume it's coming from the skin because you've had a lot of things happen to skin over the years of your life. So it's more likely, and the brain just assumes it's coming from the skin and not from an organ. So it's very difficult to understand your brain to where that actually is coming from. And you can kind of see here, that's why if you feel skin pain over here, there's something wrong with your heart. Liver, gallbladder is going to be kind of over here, also all the way up in your neck. Your lung and the diaphragm can also be neck and shoulder. So by memorizing this, you can kind of understand where different organ pain is going to be coming from. And the one that's always kind of interesting, stomach pain is all the way up here, almost where the heart is. So you're like, oh, I got stomach pain. But most of the time, small intestine is going to be down here where you think your stomach should be. Colon pain is down here. And then all the rest of this stuff is different parts. Luckily, the appendix is kind of where your appendix is for most people. So if you just take a quick look at this, this is a good way to use in a diagnostic fashion in the hospital. Let's look at some chemical senses. We're going to go into taste and smell. So how does taste and smell receptors work and how are they stimulated? And what are the pathways? Where do they go in the brain? So gustation or taste, sensation that results from the action of chemicals on taste buds. 4,000 taste buds, mainly on the tongue. Sometimes they're inside the cheek, sometimes they're on the soft palate, the pharynx or the epiglottis. This is kind of interesting because the pharynx and epiglottis, soft palate, back of the, right around the top of your tongue. If you're wondering why you can cry and you know that your tears are salty, it's because you still have taste buds on your pharynx and epiglottis when you're swallowing your tears that go down the tear duct and down the back of your throat. So you can still taste it even if you don't lick them up off your own face. We have a couple different types of lingual papilla. The filiform have no taste buds. And the filiforms are going to be these little things that stick up and they're just going to be like little fingers. The folate also have no taste buds. And if you look at your tongue, they kind of look like stretch marks on the side of your tongue. They're weakly developed in humans. Other animals have more folate papilla than us, but we still have the remnants of them on the side of our tongue that kind of just look like stretch marks. Fungiform is at the tips and sides of the tongue, and you can see the fungiform because they look like little mushrooms that grow all along your tongue. And then the valate is going to be at the rear of the tongue. It's called the circumvallate or the valate. But if you look at the back of your tongue, it looks like there's an upside down V, and that's your circumvallate. And the circumvallate contains 1/2 of all taste buds. Fungiform has a couple more. But for the most part, all those taste buds are going to have something pass by them. Same thing when we do smell. Something has to pass by that receptor for it to be turned on. So to be tasted, molecules have to dissolve in the saliva. And then they have to flood the taste pore. And you have 5 primary sensations. Salty, you need to have sodium and potassium. You need to have chloride in your body. So if you ever crave salty food, it's because you crave those ions for osmolarity in your blood. Sometimes you crave something that's sweet, like a carbohydrate, a starch, some sugars. The reason why you want sweet is because you want something with high caloric value, because you want that glucose to help with energy, and you need quick energy. Sour can be good or it can be bad, but sour, sometimes there's specific acids we want, like vitamin C in citrus fruits. We're craving those vitamins, we're craving those special types of acids that we want to have into our body because we don't have enough, or our body's telling us we need to have something sour because we need to have this particular molecule into our body. And sometimes sour can be bad too, and that's why sour and bitter usually go hand in hand. Sour and bitter can also tell us, hey, this might not be good for us. Let's spit it out. So sometimes bitter food, spoiled. Sometimes nicotine, caffeine, quinine, morphine. We might be wanting some of those, but for the most part, bitter is something that we want to try to keep out of our digestive system. And finally, this one came out. They theorized this one, decades ago, but they finally added it, and this is umami. Umami is that taste of amino acid. So if you want to have amino acids, chicken or beef, or if you want to have specific types of food, specific types of tofu, stuff like that, you can also add the umami flavor. And it's because your body lacks some amino acids, and you need to replace those amino acids so your body can get back to work of making proteins. And the taste is influenced by texture, the mouth feel of it, also by the aroma, because the food, you smell it, you see it, so all of these things are going to influence how something tastes. If you've ever gone to a restaurant, they bring you, let's just say, a fish. It doesn't matter what fish, but it looks beautiful. It smells amazing. Maybe they add some citrus, some zest of the rind on it, and it smells amazing because it's heat. It's releasing all those aromas. You smell it, you see it, you taste it. You're gonna have a memory that says that was one of the best meals you've ever ate. So if you have one of those memories, that's why all those things can be put together with taste. A hot pepper or very spicy food, you don't have a taste bud for it. That's going to stimulate a free nerve ending. So a free nerve ending is going to be a different type of sensation where it's painful, but some people over time, they like that pain. Other cultures and stuff, they don't eat hot food because they want to feel pain. They're eating hot food because it makes them sweat. So a lot of cultures that live in hotter environments, they eat hotter food. It makes them sweat. It cools them off. They've been almost taught over the years that if they eat this hot food, they'll feel cooler. And then plus that pain is going to release endorphins also that make them feel better after the meal. And you have regional differences of taste sensation on the tongue. The tip is most sensitive to sweet because you want to have sweet right away, and sometimes some of the sugars can start to dissolve right away. Edges salt and sour, and the rear is bitter. That's why sometimes you might have a gag reflex if you eat something that's bad. You start to spit it up because the bitter is towards the back. Some some cranial nerves that are involved in taste. Facial nerve, it collects the sensory information from the front 2/3 of the tongue. Glossal pharyngeal, the posterior 1/3 of the tongue, and the vagus nerve, palate, pharynx, and epiglottis. So these three are involved in all three sections of where you have taste buds. Front 2/3, back 2/3 of the back 1/3 of the tongue, and then pretty much the throat is going to be the vagus nerve. All the fibers are going to reach the solitary nucleus in the medulla oblongata, and then from there the signal is sent to two destinations. One goes to the hypothalamus and the amygdala, and that's going to control autonomic reflexes, salivation. So if you need to have more saliva, gagging and vomiting, they're going to be in the hypothalamus and amygdala. Then you have the thalamus, and that's going to relay signals to the postcentral gyrus. Postcentral gyrus is going to be chewing and muscle control, and also for conscious control of taste if it's further in front in the frontal lobe. Now we're on to the smell. So we already talked about smell. It's going to be phasic. So what happens, these little chemicals, you can call them pheromones, you can call them scents, you can call them whatever you want, but these little dots have to pass by these nerve endings to actually signal an action potential to the brain. Another thing, remember that the sense of smell is going to be phasic. So once new scents or smells come by, that's the only thing that's going to send the original action potential. And then once, even though that scent is still coming through the passing by these nerve fibers, you're not going to feel anything, or you're not going to sense the smell of anything new until a new scent. So you can put a bottle of cologne on yourself, and after a few minutes you won't smell that you have anything on you. And there's 10 to 20 million olfactory cells, but the main thing is these chemicals have to pass by. And the olfactory cells are neurons. They use chemoreceptors for chemicals. They're shaped like little bowling pins, and you can kind of see them down there. The head bears about 10 to 20 cilia, and then those little cilia are going to be down there. And what the cilia do is they just kind of move stuff along. And then pheromones and other smells stimulate them. And you can see when you breathe in, those little scents have to come in contact with that mucous membrane and also those little hairs. An only neuron in the body that's directly exposed to the external environment. And because they're exposed to the external environment, where you can see, yes, there's a little membrane of mucus right there, but for the most part, this is right in your nasal cavity. They're exposed to the external environment. So they have a very short lifespan of about 60 days where they have to continually be reproduced. And the basal cells are going to continually divide and differentiate into brand new olfactory cells. So every 2 months, you have brand new sense of smell. You have supporting cells, and then it's the basal cells that are going to be doing the dividing. The action potential then travels to the brain. The olfactory receptors can adapt very quickly. This is due to synaptic inhibition in the olfactory bulbs. All that's telling you is what is phasic nature of the cell. Once a new smell comes by, they adapt very quickly, send a signal to the brain. Something new is out there. It can be dangerous. It can be good. It can be, it can be all these different things. It can be, well, I've smelled that before. Somebody's cooking something good. You might want to go find out what that is. But if you sit there too long, you might not smell that again because your brain is now moving on to something else. And that's the phasic nature of smell. Just new stuff. And some odorants act as nociceptors of the trigeminal nerve. And nociceptor, if you remember what that term means, it's pain. So you smell ammonia, menthol, chlorine, capsaicin of hot peppers. You almost get shocked. You almost get like, oh my gosh, what's that? That's that pain receptor being activated. And now, they're tufted mitral cells, the axons, form the optic tracts. They reach the primary olfactory cortex in the inferior surface of the temporal lobe. Then they have a secondary destination in the hippocampus and amygdala, hypothalamus, insula, orbital frontal cortex. And this is where you're going to identify odors, integrate smell with taste, perceive flavor, evoke memories and emotional response and visceral reactions. And this is because hippocampus is memory, and most of the emotion, basic emotion, is going to be in the amygdala. We've already talked about when we talked about the brain. So when you're kind of looking at these, taste, smell, all of these things can actually be tied to a specific memory. You might even remember growing up that if you had a different house, it had a specific smell. And that's all because of how these connections in the brain go. And then finally, the fibers reach back to the olfactory bulbs, where granule cells inhibit the mitral and tuft cells. And that means this is going to be why odors change under different conditions. Food smells more appetizing when we're hungry, because there is that feedback where when you're hungry, you're going to smell something cooking on the grill and actually start to salivate and get very hungry. Where if you're not hungry, you smell something, you might want to leave because you don't like the smell of smoke. And all of this leads to the sense of smell does not pass through the thalamus. This is one of the major ones in the brain, or major senses, that does not go through the thalamus. If you remember the thalamus, it's a relay station. It actually controls some of the input. But because sense of smell is relatively old on our senses, on stuff that we've sensed for millennia, then we don't have to go through the thalamus. We don't have to use the thalamus sending it to different parts of the cortex. All we got to do for the olfaction is just make sure that when it comes in, we can relay it to the hypothalamus and the amygdala, primary olfactory cortex, and then send it back to the orbital frontal cortex, and then also send it back to the actual olfactory bulbs. So it has a little circuit that's a lot more compact and doesn't need to involve the thalamus. And that concludes our first micro lecture on the final chapter of this class.